Welcome. In quick summary, let's understand the transcription. Transcription we would understand in two forms. One is in prokaryotes and other is in eukaryotes. We'll be covering both of these topics separately. So to begin with the transcription in prokaryotes, the first thing that we must note is that transcription, since it's prokaryote, they don't have well-defined nucleus. Where would transcription take place? It would take place in the cytoplasm. Now what exactly is transcription? So basically the copy of genetic material from the template of DNA strand to the RNA would be called as transcription, and in the case of prokaryotes it is polycistronic. However, in the case of eukaryotes, it is monocystronic. Polycistronic means it has an ability to encode more than one polypeptides, and in the case of eukaryotes, the transcription would take place in nucleus. So another major difference. Now, when we talk about transcription, there are two strands. One is the template strand. Template strand runs from 3' to 5'. And that's what is the polarity. Now towards the 3' is the transcription initiation region where we have the promoter. Towards the end is the termination site, which is called as the terminator or the termination sequence. In between is the RNA coding sequence, which is known as the structural gene. Now against it is the antisense strand, and that is from 5' to 3', and this is also known as the same strand which is for the RNA synthesis. Now for the RNA synthesis, what is required for RNA synthesis? As we know, RNA polymerase would be required for initiation as well as elongation. Along with the RNA polymerase, in the case of eukaryotes, you would also require enhancers, which we would understand when we will be focusing on eukaryotes. So three phases. First is the initiation. The second phase is the elongation. And the third phase is the termination. Let's understand those one by one. This is the first phase. So in the first phase, what is important? The sigma bond would act as a promoter. Now here we have the RNA polymerase and a closed promoter complex. So in the beginning, you have -35 region. This -35 region is also known as the recognition sequence. Okay, and then we have the -10 region. This -10 region is known as the Pribnow box. So the sequencing for the Pribnow box would be what? The sequencing would be TATAAT, and this is known as TATAAT box. The other one, the recognition one, the sequence is TTGACA. Okay, so that would be the sequencing in the -35 region. Now, what is very important? In the initiation, what would happen? The RNA polymerase recognizes the promoter where? At the -35 region or the recognition sequence. Now, this is where it binds fully with the promoter. The next phase is the -10 region. Here the DNA unwinds. So as you can see in the previous bands, the DNA runs like this. Okay, but in the -10 region it unwinds, and now when it unwinds, it gets correctly oriented for the transcription to take place, and therefore this phase is known as the initiation phase, and this is the initiation nucleoid, which is 5'pp. Now, the next stage after it is the elongation stage. During the elongation, the RNA lengthening would take place, and this RNA lengthening is synthesized by what? It is synthesized by the DNA polymerase, and it is read as the DNA triplet code, which could provide signal that actually binds with the transcribed end, and therefore elongation is the next phase. Another important thing is only one type of RNA polymerase is involved, which is 2-alpha beta dash omega rho, and that's the only polypeptide or the only RNA polymerase which is required during this time. The last phase in the prokaryotes is the phase of termination. So here we have the termination sequence. Now termination may or may not occur or may not require the sigma factor or the rho factor. Now mRNA would not undergo any processing and may undergo translation before completion of the transcription. So translation can occur before transcription. That's extremely important in the case of termination. So here translation may occur before the completion of the transcription as both the processes take place in the diastole, and this is from 5' to 3' direction. That's one thing that is important to note. Now in most of the templates you would see 5' U A 3' and that's the sequence that it would run, and it would allow the nascent RNA. So the function of the nascent RNA is again important, and this would result in dissociation. So dissociation and termination of the transcription of RNA molecule would take place, and that would determine the final process of transcription. So three phases which are involved: initiation, elongation and termination. Since it's in the prokaryote, it would be held in the cytoplasm. The template strand would run from 3' to 5', antisense strand from 5' to 3'. The path for the antisense strand would be the same for the RNA polymerase, and that's the basic idea. And again, this is polycistronic, that is more than one polypeptides. It encloses more than one polypeptides. However, in eukaryotes it is monocistronic, that is just one polypeptide which is enclosed.